Hello, this video is here to help you do the calculations for experiment number 5, analysis of an acid-base indicator, associated with Daniel Harris's Quantitative Chemical Analysis textbook, 9th edition. So we're going to start off with what we have here. So this is just the class data that you guys input into the spreadsheet. So I've replaced your names with generic Team 1 through Team 5 names because I'll be posting this to YouTube in case it might help out other people as well. All right, so this is the raw data, and so what we have here then are for each team, we have a number of trials, and this is your data associated with each trial. This is the molarity of the hydrochloric acid sample that you titrated using these different indicators. So what we would like to do now is analyze this data and see if there are any differences between using the different indicators. Okay, so the first thing that I'm going to do is clean up this data a little bit. So I want to make sure that we're using the same label for all of the indicators. So I'm going to use the 1 or 2 letter labels that are used in the Harris book. And so those are shown in your instruction sheet. And they're here for the 5 different indicators: bromethymol blue, methyl red, bromcresol green, methyl orange, and erythrocene. So instead of using the full name, I'm going to go ahead and replace these with just the 1-letter labels. So bromcresol green was BG, erythrocene is E, and everything else looks good. Okay, now what I would like to do is sort this table so that all of the data referring to one indicator is kind of together. So what I just did there was click and drag to select that data, and then I'm going to go up here to the Sort and Filter button on let me see if I can scroll over a little bit for you. No? Okay, anyway. So it's over here. If you keep going along the top ribbon on your home menu, after you get to this section that's called Editing, there's a little button that will say Sort and Filter. So go ahead and click on that, and we're going to select the option for Custom Sort. All right, so go to the pull-down menu and select the Custom Sort option. Okay, so we're going to sort by this indicator label right here. So that's this one right here. And so what we're going to do is we're going to sort by indicator. And what we want to do is sort on the cell values. And so it's going to sort these, and it's going to sort them A to Z, so alphabetically. So we're going to get the indicators listed alphabetically. So we could add extra layers here if we wanted to sort them additionally alphabetically by team name or number or something like that, but we don't really care about that. So I'm just going to sort them like that. Boom. Okay, so now you see it's gotten rid of some of those blank rows, and now we have all the bromethymol blues, the bromcresol greens, the erythrocenes, the methyl oranges, and the methyl reds all kind of together in one section of this spreadsheet. Okay, so that looks good. So moving on. So what I'd like to do is average the results for each team's data for that particular indicator. So I'm going to create another column over here and call that average. And I'm going to go ahead and give that a unit, right? So we're measuring this in molarity units. So there we go. I'm going to give us a little bit more. Well, it already automatically wrapped the text. You can click this whether you want it to wrap the text or not, and it automatically copied the formatting, so it made it boldface. So there's our average molarity. We also want the standard deviation, which also has units of molarity. We want the percent relative standard deviation, so the percent RSD, that's a percentage, so it doesn't have units. Next, we're going to want to calculate the 95% confidence interval. So I'll abbreviate that 95% CI period, and that's also going to have units of molarity. All right, so now we've got all of those labels up there, so now we need to do those calculations. So first, for the average, I'm going to type in a formula to do the average. So I'm going to type equals average, AVERAGE, or it'll start predicting what formula you want to type in, and so you can just double click on the formula once you see it. Now, since some of these team entries have 7 trials, I'm going to go ahead and select all 7 rows right across here. So I select the cells that I want to average. Now these two that don't have numbers in them, they're not going to be included in the average, so I don't have to worry about that. It's not like they get counted as zeros or something like that. So we're going to close our parentheses and hit enter, and there's our average molarity. So we're going to do the same thing with these other quantities. I want to calculate the standard deviation. So I type equals. My formula for standard deviation is STDEV, sorry. And we want the one that's STDEV.S. That's a sample standard deviation. So STDEV.S. Then we want to select the numbers that we want to average. So I'm going to select this whole set of columns right there. So C2 through I2, close my parentheses, and so again that will calculate the standard deviation of anything that's a number. It ignores any blank spaces. So I will type enter, and there's our standard deviation. So remember, standard deviation is telling us something about the precision. And so next I'm going to calculate the percent relative standard deviation. This is useful if you just want to compare numbers that have different sizes or different units, different scales, then you can compare them. We could compare the standard deviations directly, but we're going to go ahead and do this extra one. So remember, to calculate the relative standard deviation, you need to take the standard deviation, divide it by the average, and multiply that by 100. So I'm going to type in a formula to do that. So equals the standard deviation value, so I click on that cell. Then I want to divide, so that's the / key, by the average, so I click on that. And then I want to multiply, so multiplication is the asterisk key, and you might have to type Shift 8 to get that, so it's above the number 8 on most standard keyboards. So times 100, and that will convert it into a percentage. So there's our percent relative standard deviation. So how much do these numbers vary among one another? They vary by about 1%, about 1.25%, so 1.23%. All right, so last we want to calculate our 95% confidence interval. And so the formula to do that is t that you get off the t table, so that's the t statistic, times the standard deviation divided by the square root of the number of trials. So I'm going to enter a formula to do that. So equals, so the first thing that I need is the t statistic. And so I'm going to have Excel look that up by using the built-in function TINV. So that gives you the inverse of the student t distribution. And so the first argument to that is the probability. So I want to be at the 95% confidence level. So there's a 5% chance that I'm wrong. And so the probability that we want to give it is that 5% expressed as a decimal. And so that's going to be 0.05. So 0.05. So we enter that, and then a comma. Then we need to tell it the number of degrees of freedom. All right, so that depends on how many data points we have. And if we look at this list, each row seems to maybe have a different number of data points. So I can't use the same number each time. So what I'm going to have Excel do is count how many data points we have in each one of these rows. And so to do that, I'm going to use another function. So one thing that's cool about Excel is you can put one function inside another function. So I'm going to put a function inside this TINV function. And that function is count, C-O-U-N-T. And all it does is it counts the number of cells that contain numbers in a particular range. So I need to give it a range, and so I'm going to give it all of these columns here in the first row of data. And so it's just going to tell me how many of those cells have numbers in them. In this case, that'll be 5, right? All right, so that's not quite the number of degrees of freedom. You'll remember it's the number of data points minus 1 is our number of degrees of freedom when we're looking up a number in a t-table. All right, so that big formula there, TINV, and then in parentheses 0.05, that selects the 95% confidence interval, and then the count minus 1 gives us the number of degrees of freedom. So there's our t statistic. So remember, the formula is t times s. S is the standard deviation. So I need to multiply now. So there's my asterisk. And I need to multiply by the standard deviation. But we already have a column for that. It's here, K2. So I'm just going to select that cell. Good. And then we need to divide by the square root of the number of trials. So I need a function to do the square root. So in Excel, that's SQRT. Oh, by the way, I'm inputting all these formulas as capital letters, but you can use lowercase letters too. Excel does not care. So SQRT, you can follow along if I run off the screen here. You can see what I'm typing for the formula up here in the formula bar right here. So take a look at that as well. So SQRT, then I open my parentheses, and then I need to take the square root of a number. So that square root is the number of trials, but again, because I have a different number of trials in this data set, I'm going to go through and have Excel count those. So I'm going to type another count, C-O-U-N-T, and another parentheses I need to open, and then I'm going to select again this first row of data and have it count the number of things in there. So if you notice, I just closed the parentheses on my count function, so I have one more parentheses that I need to close, and that's the one on the square root function there. All right, so there we go. I have all of my parentheses, all of my formulas, and so if I hit enter, that should give me the 95% confidence interval. Boom. There we go. All right, so you'll notice that the 95% confidence interval is close to the standard deviation, but it's a little bit bigger, right? We've had to make it a little bit wider than the standard deviation. And this is telling me that there's a 95% chance that the true molarity is within 0.0015-ish of this number, 0.09703. Okay, now we want to do that same calculation for all of the indicators, and here's the magic of Excel. So what I do is just select all of these cells that have the formula, and right here in the lower right-hand corner, there's this little green square, and if you click on that and drag down, it will create a little box for you, a selection box, and when you release the mouse, suddenly it will paste in all those formulas. And those formulas automatically update for the row that you wanted to use, unless you told it not to by typing $signs in front of some of these references. So it's automatically gonna do everything correctly for you. All right, so let's take a look at some of our numbers here. So percent relative standard deviation, that gives you a good idea of by how much the data vary from each other. So you see that at best was this set of data down here where you have a standard deviation about 0.8%, 0.9%. So that's really pretty good. So 1/10%. We might hope to get all the way down to about 1/10% or 0.2% relative standard deviation if we were really good titrators. But this 0.9 here is not terrible. But then we see that there's a 3% and a 7% and 147%. What's up with that? Well, that seems like there's a problem. This data really varies a lot from each other. Let's go take a look at the data. So we've got 0.09, 0.09, 0.09, 0.0957, 0.96. Wait, what happened there? Well, so either this is an outlier, or more likely, this is a typo. And so let's assume that it's a typo and go ahead and correct it and assume that the student that entered this really meant for there to be an extra 0 in there. So I'm going to go ahead and put an extra 0 so that it looks like the other numbers and is 0.09652. And so you can just click the check mark there and notice that it's updated our standard deviations and everything. And this now, the one that was 147%, is now our best, most precise measurement right there at 0.3. So about 1/3%, a little over 1/3%. So not bad. All right, so we've got another big number here, another 147%. Let's take a look there. Aha! Here's a cell value that's, again, 0.9. So I'm guessing it's just a typo. We're going to insert that 0. All right, good. So now at this point, you should look at some of these other numbers in one row and see if there are any numbers that seem to be outliers. So one number that seems to be very different from the other. If you think that there is, then you can do something called a Grubbs test that's described in Chapter 4 of your textbook. So go read about that, and then you can test that data point to see if it really is an outlier. If there's a 95% chance that it's an outlier, we could reject it, and then you would just delete that number from the data set. So if we have any outliers. I don't see anything that jumps out at me, but there may be some that you could check, like maybe this 0.077 here is a little bit different than these other values, and that's what's causing this percent relative standard deviation to be a little high. So that might be one we want to check with a Grubbs test, but I'm going to leave it in for purposes of this video. Okay, so now we have done our calculations on the data for each one of these teams' measurements. So now what we want to do is collect our data and create a spreadsheet that looks rather like this table from your lab handout. So the lab instructions say what we want to do is create a table like this. So we want a table with indicators listed right here. We want to list the total number of measurements over the whole class that were made using that indicator. And then we want to indicate the number of students that made that measurement. So this would be the number of not individual students, but the number of like people, analysts doing the experiment. So in our class, we have teams of several students working together to generate that data. And so this is really going to be the number of teams that contributed to that data. So were there 3 teams that measured with Brome BB indicator, or just 1 team, or 2 teams? And so we would put a number like that in here. And then what we're going to do is we're going to calculate the mean hydrochloric acid molarity averaged over the whole data set. And so if we're confused on that, this little letter A down here gives us a footnote. It says computed from all values that were not discarded with the Q-test. The Q-test is just the same kind of test like the Grubbs test. The Grubbs test is the more modern version, but a Q-test is used to reject outlying data, statistically reject it. Okay, so next we want to calculate the standard deviation over all the data from the Brome thymol blue indicator. And then we're going to calculate the percent relative standard deviation. And finally, we're going to calculate what's called a pooled standard deviation using this formula that's right here. So we're going to type this formula into Excel. So let's type this stuff in there. So I'm going to go back over here to this sheet. We're going to create a little new section down here where we do all of that. So I'm going to start here with indicator. And we're going to fill out the rest of this table. I'll leave it blank for now. And so I'll be back in just a second once I've done that. Okay, so here's our data table. I formatted it a little bit, so I selected all of these entries that I typed in, and up here I made them boldface, and I centered them left, right, and I also centered them top, bottom in each cell, so they're kind of centered in there. I thought that made it look nicer, but do whatever you'd like. Okay, so now we're ready to enter formulas to calculate all of these things. So for number of measurements, again, we want to use the count function to do that for us. So I'm going to type equals count, and we're going to count all of these cells that have data values in them. Okay, so for BB up here, here's the BB, and so we're going to select all of the BB cells. So you can click and drag any rectangular region, and this is why I grouped them together, because it makes it easy to do this. So there's still some empty cells here, but that's okay. Excel will not count those because they don't have numbers in them. So when I close my parentheses, boom, that tells me how many total measurements we made with the BB indicator. So we want to do the same thing with each of our indicators. Unfortunately, we can't copy this cell down because these are in a different order and there might be different rows. So we're going to have to do each of these by hand. So I will just do that, counting in these formulas each time. So for the methyl red, I select this block. So the brome cresol green, I select this block right here, so we only had the one set. For the methyl orange, I select these 2 rows right here. Notice that there's some empty cells, gaps in between, but that's okay. It will only count the ones that are there. And finally for the erythrocene, type count and select all of the data for the erythrocene block. There we go. Okay, so now I have all of the data counted. And so the number of students, so this is going to be the number of student groups that measured these. So for the BB indicator, it looks like I had 2 student groups that measured that. So I'm just going to enter that number manually. So 2 for that. For the methyl red, it looks like we had 2 teams that measured that. For the brom cresol green, it looks like we just had 1 group that measured that. Methyl orange, we had 2 groups. And for the methyl red, again, 2 groups. So now we want to calculate the mean HCl molarity. So again, average, and we're going to average all of the data in a block. So again, we're going to have to go through each one of these and select the data in a block. I suppose I could have, well, no, I don't think that would work either. So anyway, we're going to select all the data in a block, calculate the average each time. So equals average. We're going to select all the methyl red data, so that's all down here. We select all of this stuff, including the empty spaces. You know, you don't have to do that, but whatever. Okay, it will ignore empty spaces when it does the calculation. So average for the brom cresol green, and so that's just this data, so that should be the same as the, ooh, I made a mistake. Let's try that again. Nope, still an issue. Oh yeah, I can't spell. Average. Fabulous. All right, so now we're ready to go. Select all the data. You can just hit the enter key after you've done that, and it will insert that. We want to do methyl orange. Average. Here's all the methyl orange data, so I select that, hit enter. And finally, the erythrocene data. So equal average. Select all of the erythrocene data. There it is. And hit enter. So that gives me my mean HCl molarities. We want to do the same thing for the standard deviations. So I will type in the first formula, and then I'll cut away and come back when I've finished typing in the rest of these formulas. So equals STD, we want the standard deviation sample standard deviation. So we're going to select all the BB numbers right here, both, all the numbers from all students. Enter, there we go. And then we need to do that for each one. So I'll come back when I'm done with all of that. Okay, so now we've got all of the standard deviations computed. So now we'll calculate the relative standard deviation. So that's the same formula that we used before. It's going to be the standard deviation, so I can click on that cell, divided by the average, so click on that cell, times 100, so times is our asterisk, 100. There we go, and hit enter. Good. Now this is a case where we just use these numbers in our formula, so we can copy and paste this formula down the row by grabbing that little green square like we normally do. Okay, so there we go. So let's take a quick look at this data, and then we'll type in that big long formula, and I'll show you how that works. So first off, we see that the HCl molarities, that some of them are the same. These first 4 numbers look pretty close. This one's off by a little bit, and this one seems kind of different. So all of the numbers are different. Some of them only differ by a little bit, and some of them are more different. Again, we see the same kind of thing going on with the standard deviations. So we see one that's the most precise measurement. That's with the brom cresol green. Of course, that was only one group, so we didn't have to worry about group-to-group variation. Then the other deviations are all pretty close, except for the erythrocene. And we can see that even more clearly when we look at the percentages. So we've got about 2.5%, 1 1/3%, about 1/3%. Again, 1 and a little more than a half, 1 2/3%. And about 7%. So that one's pretty big. So not only is the erythrocene molarity different looking than the others, but the standard deviation also seems kind of high. All right, so let's use that special formula to calculate the pooled standard deviation. Again, that's going to have units of molarity. That's why the symbol is next to there. So let's go look at what that formula is. Okay, so the pooled standard deviation for capital S students, degrees of freedom is going to be n minus capital S, computed with this equation. So the 1s and the 2s here refer to the different student groups. And so S1 would be the standard deviation that comes from group number 1. N minus 1 is the number of data points for group number 1. and so on. And then we have n minus s. So we actually have that data in the top section. So let's go and enter this formula. So we want to say equal sign, and then we need to take the square root of a whole bunch of stuff. So that needs to go on the outside. So we're going to type in the sqrt function and open a parentheses. All right, so now we've sort of got this square root radical in there, and so we have to do all of this stuff. So you see that I'm going to have kind of a complicated numerator here with several terms that I'm adding together, and things are going to be multiplied and stuff. So I'm going to need parentheses around that. So sometimes Excel can be a parentheses nightmare, but you can enter formulas. That's going to be for everything in my numerator. So the first thing that I need is for the bromethymol blue, the BB indicator, for group number 1, I need their standard deviation. So that's this first row here, and I'm going to get that standard deviation right there, and then I need to square it. So what I need to do to square it is hit the caret symbol, so type your shift key and hit the number 6. On standard keyboards, the caret symbol is above the number 6. So, and I need to square that. Good. Now I'm going to multiply it by n1 minus 1. But that quantity, so I need to put that in parentheses. So I have my parentheses here. And then n1 is the number of trials that this team did. And I see have a good way of getting at that right now. So I'm going to add one more number to that. So bear with me for just a moment. I'm going to click a check mark here. It's going to complain, yeah, that I'm missing something. We're just going to click an X and go back. There we go. So what I'm going to do is create another column here and just call that number of trials. And perhaps I should have done this earlier and made my life easier. But sometimes when you're constructing a spreadsheet, it kind of goes like that. You discover a better way of doing things later on. All right, so we're going to have the number of trials here. Let me go ahead and make it boldface and, insert that. There we go. All right, so it's formatted. So now I can just type my count formula equals count, right? And then I'm going to count these number of data points right there. Good. And then I can just click and drag my little square to copy that formula down the row. And now I've counted the number of trials that each group was able to do. Okay, so now let's go back and type in that formula again, equals SQRT. Then I open my parentheses. Then I need to open another parentheses for everything that's going to be in the numerator there. And it's going to be S1, so that's group 1's data here. And so I'm going to get their standard deviation. That's cell K2, so I click on that. Then I'm going to do a square of that, so caret number 2 to square that. Then I need to type my asterisk. I'm going to open parentheses. Then I'm going to have N1, that's the number of data points for the first team's bromethymol blue data. So N minus 1. So I just clicked on the number of trials up here, and then I closed the parentheses. So there is the first term, and then I have to add one term for every student group that measured this particular indicator. But that's only 2 for us, so I just need to add another term. So plus, and plus sign separates each term here, so add another term. Need the standard deviation for this second group. Well, there it is, and I'm going to square that. Then we need to multiply that by n minus 1, so that's this number, minus 1. And then I close my parentheses there. All right, so if I had another student group here, then I would just add a plus sign and type in another term that looks like standard deviation squared times n minus 1. But I don't have another student group that measured the bromethymol blue, so we're just going to move on. So that's everything in my numerator. So I type another parentheses here to close the parentheses for my numerator. Then I need to divide, and let's go back and look at our formula, right? So I need to divide, that's this line here, by n minus s. All right, so put another one of those sheet 1 things in there, so I need to go back and get rid of that. So I'm going to enclose my denominator in parentheses, so I open another parentheses, and then I have n minus s. So n is the total number of measurements. That's in this cell right here, so I click on that, minus s. So that's this number right here, so I click on that, and then I close my parentheses right there. So that's everything in the denominator, and now I'm ready to close my final parenthesis for the square root function. It's going to take the square root of everything inside there. And I hit enter, and there we go. There is my pooled standard deviation. So this is not the total standard deviation, sort of pooled all the data together, but here we're sort of weighting each standard deviation from each student group. So that's another measure of standard deviation. They aren't too far off from each other. Okay, so it would be nice if we could just copy and paste this formula down, but sadly that's not the case because we're going to have different numbers of teams doing each one of these groups, and so we're going to have to enter them by hand, sadly. So I'll let you guys come back in just a minute when I'm done with that. So I'll cut away here while I'm... Anyway, so that gives you an idea of how to type in this one formula. So I've decided to let you guys figure out how to type in the other formulas for the other sets of data. So you just follow this one as an example. And so that will be your table of data that you need that looks like this table of data right here. So we want to create something that looks like this. We've filled in all the numbers there. Okay, good. So now we're ready to do our final set of the data analysis. So what we need to do here is look at the mean HCl molarities, and we want to pick the two that are the most different from each other. And so clearly this is the smallest one, so we want to find the one that has the maximum value. And so this is a small enough set that we can just do it by looking. And so we see that this one, 0.0966, looks like it's our largest one. So we want to compare erythrocene and methyl red to each other. So we have these two numbers, 0.09 and 0.086. Our question is, are these averages really statistically different from each other, or are they the same to within experimental error? And our experimental error, our uncertainty due to random error, we're estimating that through these standard deviations here. So what we need to do is a t-test, and in the Harris textbook this is called a case 2 t-test. And so we'll need to go through and do a t-test on these numbers. We have all the data in these tables that we need to do those t-tests, and we just have to carry them out. So I've posted some other videos on our course management system, Moodle, and so you can go and look on Moodle and find those other videos that will talk about how to do t-tests with a set of data like this, okay? So we want to do a t-test between the two that are the most different and then the next two most different quantities. So if these two are the most different, then we want to choose the next two most different. So which one is next smallest? Well, it looks like it's the methyl orange, and then we've got the BB, so the bromethymol blue. So we're going to want to do two t-tests, one between the most different and another t-test between the next most different set of indicators. So if it turns out that these two indicators are statistically significantly different from each other, then we can conclude that one of these indicators contributes some systematic error because they're giving you different numbers that disagree with each other. This is the same acid solution, so it ought to have the same molarity, but we're getting different averages, and if they differ by more than experimental error, more than random error would allow for, then there's got to be systematic error there. And so we won't know which one is systematically in error, although we have a hint that all of these other indicators are getting numbers that are more like 0.09, and this one's getting one that's more like 0.086. So maybe it's this guy, and maybe erythrocene is not such a good indicator to use for this kind of acid-base titration. So that's the end of the video. If you have any questions, please contact me and let me know how I can help out. But this is how you'll go about processing the data. So thanks a lot for watching.